Hello, I am Witold. I work at the Functional Genomics Center in Zurich, and I will be presenting the package for proteomics label-free quantification developed here. I have to assume that you know what label-free quantification in mass spectrometry is. Here is a brief slide as a reminder. I mentioned we run LFQ experiments at the Functional Genomics Center since 2007. And since 2016, we have a fully automated reproducible workflow, which also has some R components, a Shiny app, and uses some bioconductor packages. And it is available on GitHub. In 2017, we set out to improve the LFQ analysis at FGZ. The motivation for it was that there are several MS methods, data-dependent acquisition, DDA, DIA. There is a plethora of software to pre-process MS data, Progenesis, MS Fragger. There are R packages to analyze LFQ data, MS Stats, MSQROP, ROPECA. And also the experimental design sometimes is factorial and people want to test interactions. And there are more samples and conditions. Somehow we wanted to accommodate these facts by preserving MS method and experimental design specific names and figures and tables of our reports. We also wanted to make it easy to integrate new formats because we had the feeling like every month there is a new format. We wanted also to run different methods using a similar interface like the Caret package for the machine learning methods does in R. And we want to test for interactions, and when we have multiple hypothesis tests, somehow provide the integrated visualization of these results. What we did also want is to have a simple representation of the data within our package so that you don't have to learn a complex API to somehow visualize the data or implement some filtering method. And therefore, we based the whole package on a data frame, and internally the data is also represented, a single data frame. In order to achieve it, we had to provide a configuration object, which is in R6, which somehow describes what columns contain what, so the functions we implemented can work with this data frame. Although the names of the data frame are arbitrary, you can define them yourself. So we have, for instance, a hierarchy field, which points to the columns containing, for instance, the protein, peptide, and precursor IDs for proteomics, or the metabolite IDs for metabolomics data. We point to the columns containing the factors of your experimental design. We point to the column with the intensity and the raw file name. So having this, every function internally operates on a data frame, a tidy data frame, on this config object. We have predefined configuration. On the right side, you see how you could set up the analysis from, for instance, MaxQuant data. So here we read MaxQuant data, which is not in a tidy format, converted to a data frame and tidy format. Then we are adding here the annotation to this table and specify in which column the factors of this design are specified. The package contains dozens of functions, and somehow to structure these functions, some are for plotting, some are for aggregating peptides to proteins, others are for some statistics like computing CV, standard deviations of the measurement, or computing the required sample size. We came up with LFQ data class, which implements base functionality, and decorator classes, which group together similar function. For instance, the plotter class has a heat map function. You can use it to plot PCAs. And using this API, you can easily pre-process your data, transform it, aggregate peptides to proteins, but you also can create plots for your reports or store your data, write your data, your transformed data to disk. For modeling, we have implemented the same approach. All the results are stored in a tidy data frame. We support linear models, we support mixed effect models, we support generalized models. Once the models are fitted, given a model formula, you can compute ANOVA for every protein. You can store the ANOVA results, which are again a tidy table. You can visualize them. Given the models and a contrast specification, you can compute the contrasts, test the hypothesis. Using the Wald test, you can moderate the obtained p-values. Using the lemma approach, you can compute beta distribution-based significances to summarize peptide models on protein level, like suggested by the ROPECA package. So giving this API, it's relatively easy and straightforward to generate reports. Here you see an example of a LFQ report we ship to our customers. You have one section where we link to QC-related visualization and exploratory analysis-related visualization. And to the right, you see visualizations of the modeling, like p-value distribution, estimate distributions, p-value distributions of the ANOVA analysis, volcano plots. Next, you see also that we have some benchmarking facilities. So if you have a benchmarking data set where the ground truths are known, you can benchmark the functions like the normalization, protein aggregation, or the modeling functions. In conclusion, our package allows to combine normalization methods, aggregation methods with modeling and testing. Data is represented using a tidy data frame. So you can, at any point, manipulate, implement functions, link out. You can make visualizations using ggplot2. The subsystem code is relatively mature, but we work on the API. We want to use clear package patterns to make the use of the package more intuitive. The name of the package is also not fixed. You can install it using GitHub, and feedback is welcome. And last but not least, I would like to acknowledge my colleagues from the Proteome Informatics group of the FGZ, Paolo from the Proteome Service Group, and mention that this project was partially funded by the Technology Platform Fund of the University Zurich. And I would like to thank you for your attention and welcome your questions.